Cells are the smallest functional unit of living organisms, and to be functional, they need to use a certain form of energy. This energy is produced and used through the different metabolic pathways of the cell under the form of biochemical energy. Indeed, the energy used by the cell is taken from the rupture of high-energy bonds, often of a very important molecule, adenosine triphosphate or ATP. So, ATP is a primordial molecule in cell metabolism, and one could wonder how it is produced. The magic happens in a special organelle, the mitochondrion. It's in this double membrane bound organelle that one of the most important metabolic pathways of the cell takes place, the cell respiration. This set of catabolic reactions allows the production of most of the cell's ATP pool from nutrients as sugar, amino acids, and fatty acids. Most of the important reactions of this pathway being redox reactions, an oxidizing agent, also called electron acceptor, is primordial for the reactions to happen. There are different types of cell respiration, classified on the electron acceptor used in the redox chain. However, the most common one is dioxygen, O2, and in this case, we talk about aerobic respiration.
ATP synthesis and respiratory chain are coupled, one doesn't work without the other. To understand this coupling, we will use a graph of O2 consumption and ATP synthesis. If we add ADP and phosphate, we can see a slow increase of O2 consumption and no increase of ATP synthesis. Then we add succinates. We can see high increase of O2 consumption and starts of ATP synthesis. First of all, to be consumed, O2 needs both ADP and phosphate and also succinates. But why? Succinate is the electron donor and the energy comes from electron gradients. ADP and phosphate is needed for ATP synthesis. We can also see that both reactions happened at the same time. When O2 consumption increases, ATP synthesis increases too. Then we have cyanide, which is a kind of poison for O2 consumption. If O2 consumption stops, respiratory chain stops and ATP synthesis stops. If ATP synthesis stops, we will have an electron accumulation in the inner membrane space. The gradients will be so high what will lead to the stop pumping of protons, in fact NADH. Electron on chain will stop, respiratory chain will stop even if it takes more time.
Electron transport chain, through oxidation, pumps protons out of the matrix in the intermembrane space. These protons are used by ATP synthase to produce ATP, making these protons going back in the matrix through a phosphorylation process, so they are linked. Inhibitors can act on difference complex of the electrons transport chain by inhibiting oxidation, and by this process, proton cannot cross the membrane to join the intermembrane space and accumulate in the matrix. There is no creation of gradient, so it does not allow protons to flow through the ATP synthase, and so no ATP is produced. Venturidine and oligomycin inhibits O2 consumption. When it's added, ATP synthase stop and electron transport chain decrease drastically and practically stop.
What is an uncoupler? It is an agent that dissociates two integrated series of chemical reaction. In other terms, it kind of steals the protons which avoid the gradient to work. It inhibits only the ATP synthesis. There are two uncoupling products, DNP and FCCP. In this diagram, DNP is added, which produces an increase of the consumption of oxygen, but ATP does not move. It is called uncoupler because we can clearly see that the two processes are now not working together anymore. Why is it important to have uncouplers? They prevent the formation of ATP in oxidative phosphorylation in the mitochondria by dissociating two reactions, phosphorylation and the electron transport plus oxidation. Why is it relevant for us? The result of uncoupling, oxidation, and ATP synthesis is the heat production, therefore thermogenesis. It is really useful for the animals which are hibernating. There is a consumption of nutrients, but instead of having an ATP synthesis, there is heat production in the brown adipose tissues.